So we'll start with a brief introduction and go from there. Okay. So in this module, our goal is to provide some common knowledge base for all the participants. I heard that almost all of you work in microbiome one way or another, but some of you might be more familiar with the wet lab side, and maybe that's why you're here for the bioinformatics analysis. Some of you may be familiar with bioinformatics analysis, but less familiar with the rationales of doing microbiome studies. So this lecture hopefully help you balance out a little bit. We also encourage you to work with each other. The idea of in-person workshop is that you can work with the person beside you and help each other out during the tutorial session. And also feel free to stop me anytime you have questions. Okay, so the learning objective for this particular session is to learn key terms and concepts for microbiome research, understand the key advancements of microbiome study and how sequencing technology have enabled the study. I cut down some of these for brevity, but I'll go over. And we also will give you an overview of each of the modules. And again, I cut down some of the material just to compress the information a bit, given that most of you have the background already and probably would rather have more time on the hands-on and on the other sections. And lastly, we'll finish with a little bit of caveats and challenges associated with microbiome studies and a bit of a future forward-looking statement. And I should also thank Rob for some of these original slides I've been recycling for the last few years. Okay, so microbiome research aims to explore the relationship between microbes and their environment, ranging from natural environments to built environments to living hosts, including human and animals. To accomplish this, we use a series of experimental and computational techniques to make inference about the community. These include various omics techniques, and in this workshop, you'll see mostly genomics and transcriptomics techniques. And we will also learn a bit about how to set up these applications and so on in the AWS, well, not set up per se, how to run these in AWS environment. And as a note that the AWS instance that's been made available to you is also publicly available, so you can actually reproduce some of these work, assume that you have an AWS account and able to launch an instance. So the traditional microbiology view is that microbes are studied in pure culture, as we still do. And often, when we do sequencing, we need to use isolated pure culture. But as techniques improve, we know that we can start to use less and less material, and hence the field of microbiome and expansion of more direct sequencing techniques. We also know that bacteria in nature are not found in isolation, but in mixed community. And here are some stats I'm sure some of you are familiar with in terms of how rich the number, in terms of number of species or number of types of microbes, and also the number of microbial cells that are on Earth. And indeed, it's more than the number of stars in the known universe. So bacterial populations cooperate and compete with each other and interact with host and environment. And as mentioned, we want to use omics technologies to study these phenomena. So microbiome is a term that has been around, I think, since the 90s or so. And it's attributed actually to Josh Lederberg. He's not the first person that came up with the term, but he's the person that actually sort of gave a precise definition. It's a collection of genomes in our indigenous microbes. The idea being that a comprehensive genetic view of Homo sapien as a life form, a meta-organism, should include the genes of our microbiome. And the microbiota, on the other hand, describe the actual organisms, microorganisms found in that particular setting. And I'll show a diagram that gives that sort of impression. And if you're interested in the history of how these terms come about, you can actually look at this link down here. So metagenome, on the other hand, originally was defined as the advances in molecular biology and eukaryotic genomics, which have laid the groundwork for cloning and functional analysis of collected genomes of soil microflora, which returned the metagenome of the soil. And meta means beyond. So it indeed is intended to be a way of study the functions of the organisms in a community. And this term was coined by Joe Handelsman. So metagenomics now have been used to describe the study of genetic material directly obtained from a sample, usually through shotgun sequencing. So marker gene surveys, which will be the next session, focuses on the amplification and sequencing of the target conserved regions, such as the 16S ribosomal RNA gene, and rather than the random sequencing of the genetic material. So that's a differentiation between metagenomics and marker gene-based analysis. So I've found this diagram in a review article from 2020 and sort of sums up how microbiome is, the statement is that microbiome is always composed of all members which interact with each other, living in the same habitat and form the ecological niche together. And so this diagram sort of shows the different components of a microbiome, with the microbiota or the microorganisms being one component of that community. And we also know, of course, that most of the microorganisms still cannot be cultivated in isolation in the lab, and that's coined therefore the term great plate count anomaly. And even though with the newer sort of cultiome type of approach of systematically refine the nutrient requirement of organism in order to culture them in the lab, the vast majority of organisms, as they live in a complex community, is still difficult to culture in the lab. So metagenomics offer an effective but not perfect way to profile and structure the functions of the microbial community. And there are a lot of microbiome papers, as you know, and some of you have contributed to this count, but last I checked, there's over 100,000 papers in PubMed from the last 20 years or so. We also know that early on in the human microbiome projects, most of the genes found in and on us are not our own genes, but that of the microbes that are living in and on us. So the human gut microbiome consists of 1,200 different species or so, and each person, and I guess that number does change from time to time, and some of you might have an updated number to this, and each of person harbors about 200 species. And all in all, it encodes about 500 times more genes than the human genome. So that's sort of the scale that we're looking at. We have about 25,000 genes in the human genome, but roughly 2 to 3 million microbial genes that are found in and on us. And also earlier on, we know that different habitats or different niches of the human body have different communities, and this I guess an overview or summarize the different types of microbes that can be found on skin versus gut versus what else is there? Vaginal microbiome and so on and so forth. So what do they do in and on us? So they prevent pathogens from easily colonizing the host, and they're quite a recent paper that actually further describe how how the microbiome can actually retard pathogens from colonizing. And it also provide metabolic functions that are not encoded by the host. We already know that they help to digest some dietary fibers that are not digestible by our own enzyme and produce different types of vitamins. We also know that through notobiotics studies and germ-free mice studies that without host microbiome interaction, the host immune system do not develop properly. So they modulate host immunity and defend against pathogens as well. They may actually influence host behavior through reducing synaptic connectivities, and a lot of more recent papers of highlighting the long-range interactions between microbial products and the host organs. So that was mostly on the human microbiome, but there are also many more, and I just heard one here, animal microbiome studies that have been done. They play similar roles in the animal host, and manipulate the microbiomes can actually make agriculture more sustainable. For example, reducing the use of antibiotics and reducing harboring of harmful pathogens through the use of microbiome or other ways of manipulating the microbiome. And not just living systems, microbiome studies, of course, have traditionally long been done using samples from environmental sources. So many early microbiome studies look at microbial compositions and functions in natural habitats. And these activities can shape planetary and microorganism health. So they, as you know, they influence the nutrient cycles such as carbon cycles in the environment. And the different metabolic capacities are found in the different environment. As shown in the diagram here, you can actually, based on the metabolic capacities, predict the environment that the organism may be from. Okay, so the more we learn about the microbial community, the more we see that affecting one community through anthropological activities, such as use of antibiotics, climate change, deforestation, agricultural activities, can have widespread effect and that ripple through the interconnected microbiota. So more sophisticated omics platforms and the analysis therefore are needed to tease out the complex interaction. The One Health concept state that interconnection of all areas of life can be elucidated through the study of microbiome dynamics. And this review also highlighted how it's possible to integrate different data sources and different modality of data to do a more holistic study. And indeed, that's the challenge and the aspiration for complex microbiome studies to understand interactions. So just very briefly to mention that, of course, all this is not possible without the advancement of sequencing technology, especially in the next gen and third generation sequencing technologies that allow us to generate large amount of data at a reasonably low cost. And this outdated diagram, which continue to increase to date, really shows the many-fold of increase. I think it's like 10 to a 12-fold of increase from Sanger sequencing to the development of next-gen sequencing. And since 2005 or so, when the early sort of Solexa or the Illumina machines first came out, they indeed have increased throughput many folds, with the cost continuing to come down. And as you know, that with the current pandemic with SARS-CoV-2, it's indeed the first time in our human history that we actually using genomics in a global sense to track a pathogen. And that's outside the scope of this workshop, but it really shows that the sequencing capacity and the use of genomics data can help us understand the microbial world a lot better if it's used systematically. Okay, so there are different questions that a microbiome study can answer, from who is there, what can they do, to what are they doing, right? So who is there can be answered by marker gene type of analysis, by metagenomics based taxonomic classification, which we'll see in the next module and tomorrow morning. And then we might also be interested in what they can do in the community. In other words, the use of genomics shotgun sequencing can tell you what kind of genes are found in the community, but it doesn't necessarily tell you if the organism is actually actively expressing those genes, or if the organism is indeed alive or active. So in order to understand what are they doing, you need to be looking at the expressed genes or the proteins. And on day 3, we'll see a bit about the transcriptomic type of study. What we won't be covering is metabolic studies and metabolite analysis and proteomic analysis in this workshop. So well, this slide is really just to say that there's a lot of experiments that we can, especially the one challenge with microbiome studies, that it's usually very difficult to replicate the experiment or to resample a particular environment or host, and you won't necessarily get the same thing back. So so more and more studies are looking at using animal models or using in vitro conditions to try to understand the complex interaction. And indeed, the IMPACTT consortium that sponsored this workshop, and many of you are part of that consortium, indeed systematically established mouse models and cultural collections to allow more reproducible study and allow scientists to manipulate the experimental parameters to try to understand the interactions. So the long-read so-called 3rd generation technology have also become better and better. And as a result, we increasingly apply long-read sequencing technologies to samples to try to get the contextual information of a gene. So as you know, if you chop up the genomes into small fragments and sequence on the short-read sequencers, you need to assemble those reads in order to see what's adjacent to those genes. And you will see some of that in the module 5 or the morning of the 3rd day. But the long read enable more taxonomic signals and also functional predictions to be done directly on the sequences and minimize the impact of misassembly and other artifacts. So the trade-off is that of course, the long read technologies are still have lower accuracies than the Illumina platform. So there are still some outstanding challenges and opportunities for microbiome studies. And first, it's clear that we need to move away from cataloging the microbiome to a more mechanistic understanding of the interactions between the host, the environment, and the microbiome. The diversity or the microbiota, I should say, the diversity and highly uneven and rapidly evolving nature of the microorganisms means that fully cataloging the microbial diversity is not possible. And even trying to understand the diversity of a single species or single organism is very challenging, right? We've been studying E. coli for over a century now, and we still don't know exactly what an E. coli is, right? The core genome of E. coli only represents about 5% of the total possible genes that we found in a so-called E. coli bacterium to date, right? So to identify the patterns and the principles governing the interactions these are the important exercises. So as mentioned, the use of animal models and the use of time series type of studies, the use of in vitro studies will allow us to better dissect these type of interactions and to develop better statistical tools in analysis. And you'll see a little bit of that this afternoon. And by using animal models, we're able to understand and manipulate the intra-host signaling from the microbiome. So it is important to understand that the resilience of the microbiome and its ability to withstand disruptions through a One Health lens, again, trying to untangle the complex interaction. What else did I? So, and some of our goals indeed is to understand with all these diversity, what constitutes a healthy microbiome, what's a stable composition for a community, and to develop microbiome-based clinical intervention based on what we learned about the patterns and principles of what constitutes a stable community. Okay, so very briefly, these are the list of modules for the rest of the workshop. So today we'll go over marker gene analysis with Rob and Diana. And in this afternoon, we'll hear a bit about sample processing and sample size calculation from Claire and Anita. And on tomorrow, it's all Morgan and Robin will be covering metagenomics data sets, first doing taxonomic analysis, trying to see who's there, and then carrying out functional analysis to see what are the capacity of the community. And on day 3, you'll hear about metagenome assembly and metagenome assembled genomes, so how to provide better context using better gene context using metagenomics data. And this will be with Laura and Nikhil. And then on the afternoon of day 3, you'll learn about metatranscriptomics with John and Anna. So I'll just finish with some concerns with microbiome analysis. So first, as I mentioned, there is still data quality issue, especially when sequencing error can be confounded with rare strains that are found in a sample. Sometimes it's hard to untangle those sequencing errors versus rare strains. And there's also still issues, especially with some of the target amplicon protocols. You can have chimeric sequences, and if you have a sample that's that doesn't have a lot of material, then and amplification artifacts and cloning of restriction fragments are some of the issues that can create chimeric sequences in your data. And as I also mentioned at the beginning, contextual information, in other words, metadata, is important for the interpretation of sequence data, and the acquisition and indeed the availability, even if they're acquired by the researchers, the sharing of such data is often not often an afterthought. So as we're studying a complex ecosystem, the amount of contextual information that we need to think about and collect, it can be quite daunting. So trying to tease out which ones are important for your study is still work in progress. And mentioned the public health data set often lack the proper metadata to allow you to reuse the data set. So there are some microbiome experiments, and also building specific fields for the specific environment and specific study types. And this work is still very much ongoing, and my group, for example, is involved in some of these to make sure that in the public repositories, the data that the metadata that are reported are consistently reported, and also using standardized vocabularies. Okay, so lastly, as I mentioned, reproducibility is still an issue in microbiome studies. And early days, and I think Rob will talk about some of these, is that to use the different 16S hypervariable regions, you can actually get different results in terms of the community profile, because the different primers have differential amplification or preference for different species. So there are also differences in sequencing platforms and sampling conditions that can affect your results. I was involved in the early days of the human microbiome project, and one of the activities that I was peripherally involved was just looking at the different extraction kits and how they can impact the readout, right, the sequence that I generate at the end. That type of issue has generally been much better or well described in literatures nowadays, but it still remains an issue that the different experimental protocols and different sequencing protocols can affect your data set, and part of the metadata capturing therefore should include how you generated the data. And lastly, workflow, in other words, what bioinformatic tools you use to do the analysis can also affect the comparability and reproducibility of the different studies. And indeed, it's very difficult to predict the impact of using different tools. And there are many benchmark type of studies that are done, but the key lesson here is try to record and capture as much as possible the workflow, the tools, the version of the tools that you use to help to improve reproducibility.